Hello, I'm Lars Jeuken, reader at the University of Leeds. Today we're going to see how the shape of an enzyme is important for its function. To fully explain how enzymes work, we also need to understand the chemistry. However, as not all of you will have taken chemistry at A levels, this podcast will not describe the details of the chemistry. If you're interested in the chemistry of life, perhaps you should think about studying biochemistry at the university, such as the University of Leeds. Besides discussing how enzymes work, I will also say a bit on how we study enzymes, and I will spend a bit of time talking about what the future might hold in our research of enzymes. In this first of two podcasts, I will focus on a single enzyme called lysozyme. Lysozyme is an excellent example for this podcast for both historical and tutorial reasons. Historically, lysozyme is important because it was the first enzymes for which we knew the shape. However, in science, we do not talk about the shape, but instead we talk about the structure of an enzyme. When we work hard and finally know the structure of an enzyme, we often say we have solved the structure. This phrase nicely illustrates how difficult it is to actually get to a structure. It's like solving a big puzzle. In 1965, David Phillips used X-ray diffraction to solve the structure of lysozyme. And here I have a model of the structure. Importantly, he did not only solve the structure of lysozyme, but used this structure to explain why the enzyme works as it does. In this way, he was the father of what is now known as the structure-function relationship in biochemistry. The structure-function relationship of an enzyme means exactly what it says on the tin. The structure of an enzyme is directly related to its function. And to understand how an enzyme works, we need to know the structure. Before I can explain why the structure of lysozyme is so important for its function, I of course have to tell you something about its function. Lysozyme was famously discovered by Fleming in 1922, supposedly when a nasal dripping fell on a plate on which bacteria were growing. Not really sure whether that's true, but it doesn't matter. The information itself tells you two things. Lysozyme is present in your nose drippings and it kills bacteria. In fact, lysozyme is present in all your mucosal secretions, including saliva and tears.